All right, today we're going to crack open one of the oldest and honestly one of the most ingenious tools in the entire medical lab. We're talking about blood agar. Yeah, it's basically a crime scene investigation, but for microbiologists, all happening in a tiny petri dish.
So, to really get why this little dish is so brilliant, you first have to understand the huge problem it solves. Think about it. Every time a doctor takes a sample, say a throat swab, they're immediately up against this classic detective's dilemma. And it's a really serious question, right? Your body is just this bustling city of bacteria. Most of them are totally harmless, just minding their own business. But hiding in that crowd, there could be one bad actor, a pathogen, that's causing all the trouble.
So, the big challenge is how do you pick out that one troublemaker from a sample that's just swarming with other microbes? You can't just stick it under a microscope. A lot of the time, they all look like the same tiny little dots. You need a trick. You need a way to make the culprit give itself away.
And that's where this elegant, century-old solution comes in, the living petri dish we call blood agar. See, it's not just a buffet for bacteria to grow on. Oh no, it's an interrogation room. So what is this stuff? Well, at its heart, blood agar is designed to do two jobs at the same time. First, it's enriched. That just means it's packed with all sorts of extra nutrients, so even the fussiest bacteria, we call them fastidious, have everything they need to grow. But second, and this is the clever part, it's differential. That means it's designed to make different bacteria show their true colors, to reveal their unique fingerprints, so we can tell them apart.
The best way to think about it is like a recipe for a diagnosis. First up, you've got the scaffold. That's the agar itself, which is this jelly-like stuff from seaweed that gives the whole thing its structure. Then you've got the buffet, these things called peptones, which are basically protein shakes for the microbes, giving them all the energy and nitrogen they need. Next, the defense. You need salts, like plain old sodium chloride, to keep the internal environment just right. This maintains the osmotic balance, which is just a fancy way of saying it stops the red blood cells from popping before the experiment even starts. And finally, the star of the show, the reveal. That's the blood. This is the secret ingredient that turns a simple growth medium into a powerful detection tool. But hold on, why sheep's blood? That sounds kind of random, right? Well, it's not random at all. It's actually a brilliant bit of science. For one, it gives you these super clear, easy-to-read patterns, especially for major suspects like the bug that causes strep throat. And second, get this, sheep's blood naturally stops a harmless look-alike bacteria from growing. This little guy often hangs out in our throats and could cause a false alarm. So using sheep's blood is like having a built-in filter to reduce confusion. How cool is that? Now, here's a fun fact that might surprise you. Using human blood is a terrible idea. You'd think it'd be the perfect choice, right? I mean, it's a human infection. But the problem is, our blood can have all sorts of stuff in it, like leftover antibiotics or antibodies from a previous cold, and those things can actually kill the very bacteria the lab is trying to find, which could totally screw up the diagnosis. It's a great reminder that in science, the most obvious answer isn't always the best one. Okay, so our crime scene is all set up. Now for the really cool part. How do we actually read the clues? How do these tiny bacteria leave their fingerprints all over the place? The key piece of evidence, the smoking gun, is something called hemolysis. This is the main event. It's the visible carnage left behind when bacterial toxins, called hemolysins, go on the attack and start breaking down all those red blood cells in the agar. This is how the bad guys give themselves away. All right, first up is the most dramatic clue of all, beta hemolysis. This is like a signed confession. The bacteria here are pumping out toxins so powerful, they completely obliterate the red blood cells. What you're left with is this perfectly clear, see-through halo around the colony. I mean, you can literally hold the plate up and read a book through it. That tells you you're dealing with a seriously destructive pathogen. Then there's a more subtle clue called alpha hemolysis. In this case, the bacteria aren't blowing up the cells. Instead, they're kind of bruising them. They release something that chemically changes the hemoglobin inside the red blood cells, oxidizing it into this greenish-brown color. So it's not total destruction, it's more of a chemical attack. And last but not least, you have gamma hemolysis. And gamma is really just a fancy way of saying nothing happens. The bacteria grow just fine, but they leave the red blood cells completely alone. Now, just because there's no reaction doesn't mean the bug is harmless, but it tells the microbiologist a very specific piece of information. This organism doesn't use toxins that attack red blood cells. And in a detective story, the absence of a clue is a clue. Okay, so we've got these patterns: clear, greenish, or nothing at all. But how does that actually help us name the culprit? Well, this is where all that detective work really pays off. This is where we match the evidence to the most wanted list. That big, clear zone of beta hemolysis, that's the classic calling card of Streptococcus pyogenes. Yep, the bug that causes strep throat. That greenish, bruised-looking alpha hemolysis, that's a huge clue for its cousin, Streptococcus pneumonia, a major cause of pneumonia. And then you get something really specific, like double zone hemolysis. It's this wild pattern with two rings, a clear one inside and a hazy one outside. That is the dead giveaway signature of Clostridium perfringens, the nasty bacterium that causes gas gangrene. But, sometimes the crime scene is just too chaotic. You have so many different types of bacteria growing that it's nearly impossible to spot the one you're actually looking for. And when that happens, microbiologists have to pull out some more advanced tools. This is how you narrow down the list of suspects. On a standard blood agar plate, pretty much anything can grow, so it can look like a wild party. But then you have a selective version, like CNA blood agar. Think of this as putting up police tape at the crime scene. It's got a couple of antibiotics mixed in that specifically stop a whole class of bacteria, the gram-negatives, from growing. This clears out all the innocent bystanders, all the background noise, making it way easier to spot and question the key gram-positive suspects, like staph and strep. So, when it's all said and done, what's the final verdict on this old-school piece of medical tech? What's its legacy in our super modern high-tech world? I mean, think about it. For more than 100 years, this simple method has been a cornerstone of microbiology labs everywhere. That's just an incredible testament to a powerful idea that has absolutely stood the test of time. And this is why it all matters to you. When your doctor takes that throat swab, this is where it goes. It goes onto a blood agar plate. And the patterns that show up in just 24 hours can tell your doctor, hey, this is strep throat, or nope, it's just a virus. No antibiotics needed. That simple test is the fastest link between your symptoms and getting the right prescription, or avoiding one you don't need. It is fast, it is cheap, and it is unbelievably effective. So I'll leave you with this. In an age where we have mind-blowing genetic sequencing and all sorts of fancy molecular diagnostics, one of the most powerful and reliable detectives we have in the fight against infectious disease is still this humble century-old invention. Just a simple dish of blood and jelly. Pretty amazing, right? Thanks for tuning in.